Hello everyone, so in today's video I will discuss Pinacol rearrangement, or simply you can say Pinacol rearrangement. So let's start. So in this rearrangement, as you can see, why the name is Pinacolone, that first we have to understand. So this molecule 2,3-dimethyl butane 2,3-diol, the common name is Pinacol, and look at the product side, methyl tertiary butyl ketone, common name is Pinacolone. That is the first reaction for this type of rearrangement, and that is why based on this substrate molecules common name and the product molecules common name, this rearrangement is known as Pinacolone rearrangement. So as you can see, this is 1,2-diol, that is vicinal diol. Pinacol rearrangement is acid catalyzed rearrangement of 1,2-diols or vicinal diols. So it is not just for these molecules, they are just first reported, but not restricted to this molecule only, but wide variety of other vicinal diols also we can use. So what is happening here? In presence of acidic condition, one of the OH group will be removed as a leaving group, and that is water molecule. As you can see, this will be the intermediate, though in the mechanism part we will see in detail that this type of fully formed free carbocation may not be formed, but just for the sake of understanding, as you can see, one OH is removed as a in the form of water molecule because it will be protonated in presence of H plus, and here we are getting some carbocation. It may be free carbocation or carbocation like character, and then one of this methyl group is shifting to this position, and finally we are getting this type of ketone. Okay, so that is all about this rearrangement, and next we will go to the mechanism. So in the mechanism part 1, as you can see, we will generate a carbocation intermediate. Now what is happening first? There will be protonation. So one of the OH, this is a symmetric molecule, so we don't have to think about which OH you will choose. Just simply protonate and it will be converted to OH2 plus. Now it is a good leaving group, right? So there will be removal of this leaving group and you are getting this type of carbocation. So we have already generated the carbocation intermediate. Now in part 2, what we will do? In part 2, there will be rearrangement of carbocation, that is first formed in part 1, and that will be by the migration of one of this methyl group. So what is happening? If I am migrating this methyl group to this carbocation, the new carbocation will be generated at this position. Now what is the driving force behind it? The driving force behind it is that if you simply follow this resonance, the lone pair of oxygen is making this carbocation stable because of this type of resonance, and that is the driving force. So the original carbocation and the newly formed carbocation, just compare it, it is more stable, fine. And finally this H will be removed, that is final step is deprotonation, and we are getting our product CH3CO, and then there is a tertiary butyl group, right? Now this is for symmetric molecule. Next, okay, before that, one important point here, the carbocation formed by the departure of water molecule. So this carbocation we are getting by the departure of water molecule, and this type of departure of water molecule pulls the migrating group, that is the carbocation is generated and now it is pulling the migrating group across at the same time as the oxygen lone pair pushes it. So the oxygen's lone pair, it is pushing it and this methyl group is migrating. So this is a type of, you can say, push-pull type of mechanism. Okay. Now the question arises that is it stepwise or concerted? If you look at the previous mechanism, it seems as if it is stepwise. But a concerted mechanism is also possible where you will not get any free carbocation. So the first step as it is, one of the OH you will protonate, and then what is happening here? This lone pair, it is pushing and one of the methyl group is shifting or migrating and water molecule is removed. So finally you are getting this, and there will be obviously deprotonation. So here we are not generating any carbocation intermediate. Now the question arises whether we will consider concerted or stepwise. Now see, there is no straightforward answer, but if you want to know about it in detail, there is a good reference for that. I have already mentioned it here. So here they have done a theoretical study concerning both concerted and stepwise pathways for this Pinacol rearrangement, and they have found that concerted mechanism is possible in non-ionizing solvent. Non-ionizing solvent means which the solvents that cannot stabilize free carbocation. So then you can expect concerted mechanism, but mostly it is concerted mechanism. Fine. The next important question. Now, until now you have seen symmetric molecules. So the which OH group will be protonated, that question we have not, that is not important. That question never arises, right? But now we have to talk about regioselectivity. That means which OH we will protonate. So simply look at this molecule. This is the middle molecule, and the green OH and the pink OH, obviously their environment not same. Now if you are removing this OH, that is the pink OH, you are getting the carbocation. And if you are removing the green OH, you are getting the carbocation at the left hand side. Now obviously this carbocation is more stable, right, because of these two pH groups. So that is why the more stable carbocation will be formed at that position. That OH group you have to remove, right? So the answer is the OH group leads to a more stable carbocation. That OH group you have to remove. And finally, what is happening here? If I am simply numbering the carbon 1, 2, 3, 4, 5, 6, as if this 2,6 bond is migrating here, and you are getting now 1, 2, 3, 4, 5, 6. So you are getting a 6-member ring. 1,6 bond formed and 2,6 bond broken, and at this position you are get basically here full mechanism I have not shown. There will be CO double bond, and finally this OH will be removed, and you will be getting this ring expanded product. And this is not, it may be minor product, but this is not the major product. One more example. So just look at this middle molecule. Here if we protonate the pink OH, you are getting positive charge at this position. If you are protonating the green OH, you are getting positive charge at this position. So obviously the left hand side carbocation will be more stable. So ultimately you can say this pink OH will be removed, and this green oxygen that will be retained. Okay, and one of this methyl group that will be migrating. So that is why in this side you can see 2 pH and 1 methyl. Now migration. Which group will migrate? So in the previous example you have seen, just look at this. In the previous example, both the groups, that is this side or this side, it is same group. Similarly, in the other carbon containing OH, these two groups are same, right? So it is on one side symmetrical, another side also symmetrical. Now the molecule that we have taken, as a whole it is symmetrical, but if you just focus on one half, obviously there is two group, different group. Now the question arises, which group will migrate, right? So as you can see in this molecule, the carbocation that will be common, whether you remove this OH or this OH, it doesn't matter, it will produce the same carbocation, right? Now the question arises whether pH group will migrate or methyl group will migrate. If you are migrating the pH group, you will get the first product. If you are migrating the methyl group, you are getting the second product, right? Now here also we do not have any straightforward answer. It is not so straightforward to define unambiguous migratory aptitude that you can blindly follow always. No, that we do not have here. So this unambiguous migratory aptitude of a specific group is we cannot give, because the stability of the transition state depends on the activity of migrating group. It is true, but also it depends on the other factors such as the electronic static factor, effects of the non-migrating group substituents, that is the remaining part of the molecule, that also matters. So that is why straightforward answer we cannot give, but still nevertheless there are some general trend that can be concluded from experimental data, right? So as a reference guide you can say that you can place the migratory capacity of aryl group always greater than alkyl group, but when it is the question of hydrogen shift, they have shown mixed result relative to aryl. So sometimes you will see H is shifting, sometimes pH is shifting, though most of the time in some books you can find this type of groups, and then there is alkyl, but I will suggest you do not blindly simply follow it, because it is not so straightforward, right? So I will suggest not to follow it blindly. So as far as hydrogen shift, they have shown mixed result relative to aryl or alkyl group, and that depends on the structure of the reactant and obviously the experimental conditions. Now if aryl group is more than alkyl group, that is predominating, now obviously we have to consider this one and not this one. That is why the first compound that will be the major product. Now another example where we have a pH group and another aryl. Now aryl means this is also pH, but we have a substitution at 4 position. Now if you simply compare two aryl group, one is simply benzene ring, another one is having some OMe group, obviously the group which is having this OMe substitution, that will be migrating. So that is why if AR is migrating to the other side, you will get two AR group and one pH, right? Now we will start the different types of example. The first example you can see here, we will see some ring expansion. Though one ring expansion we have already seen, let's see another example. So here which side we will get the, that is we will get the carbocation, obviously this right hand side. So this wedge will be removed in the form of water molecule. Now there will be bond migration. So as you can see, you can either migrate the 2,3 bond or you can migrate a 2,5, both are actually same thing. So here I have migrating 5,2 bond. That means you will be getting 1,5 connection and the 2,5 connection will be lost, and at 2 position there will be C double bond O. Okay, so this will be our product. From 4 member ring you are getting 5 member ring, that is a ring expansion. Let's say another example where you will get no expansion. So it is not that always there will be expansion, it depends. So in this case, positive charge at this position, that will be more stable, because other side we have simply hydrogen. And as this is the case, now when this okay, better I do it. So when this will be pushing like this, one of these hydrogen, there are 2 hydrogen, right? So one of this hydrogen will be going this side. So that is why the ring size is still 5. Initially it is 5, now it is also 5. So in the previous case, what is happening? The ring is expanded because the positive charge you are generating on the other side, not to that side where the ring is present. But now you are generating the positive charge on the carbon which is part of the ring. That is why no question of ring expansion. So here we are not getting here ring expansion. Fine. Next example, KCP versus TCP product. So in this molecule, obviously we can understand if it is carbon number 1, this is 2, positive charge we have to generate at position 1. Because by removing this OH group, because 2 pH groups are there, it will stabilize it. Now after this positive charge generation, one of this H will be moving towards, because there is no choice, you have 2 hydrogen, it will be moving towards 1, so you will get this product. Now this product is KCP, which will be formed very fast, and this is our expected product. Now what will happen, if you keep this molecule for a long time in this acidic condition, then there will be some slow reaction with longer time. Now look at the TCP product, as if this pH and H, they have exchanged their position. Now if you simply forget about Pinacol rearrangement, just look at the 2 molecules. This molecule is C double bond O and we have hydrogen attached to it, and this molecule is CO attached to pH. Now because of some conjugation with this C double bond O and the CC double bonds in the pH ring, this molecule is more stable. So from product stability point of view, this is more stable, and that is why this is the TCP product, thermodynamically controlled product. But how it is generated when we are keeping this molecule KCP product, expected product for a long time? So what is happening here? For that you have to understand, just move from KCP to this molecule, as if in presence of H plus this OH is now protonated. Now once this OH is protonated, after resonance you can generate the charge at carbon by this breaking this double bond. Now what will happen, one of the pH, it can shift to this position. Now when it is shifting to this position, positive charge will be generated at one position again, as if, see, initially we have done so. Now once it is generated here, finally this bond, OH bond will be broken, there will be one hydrogen removal, and this hydrogen will be migrating here. So finally we are getting PHCH2CO pH. But if you simply ignore this part, and if you simply look at the TCP product, suppose you have kept this molecule for a long time, you are doing the reaction for a long time, you are getting this TCP product. It seems, if you simply ignore this part, that is fast KCP is formed, then slow time with longer time we are getting TCP. If you simply forget it, TCP product you are getting as if positive charge is generated at 2 positions. Look at the just starting material, look at it, as if positive charge is generated here, and one of the pH is going there, as if it is generated like that. But this is not the case, it is happening through this conversion from KCP to TCP. Fine. Next example, here we will get some unexpected product. Let's see what is happening here. In presence of H plus, this is symmetric molecule 5. So what we will be doing? We will be removing one of the OH in the form of water. So suppose you are converting this OH to a OH2 plus, and then you are removing water molecule and you are getting this positive charge here. But see, it is expected, but it is highly unstable carbocation. Carbocation, it is sp2 hybridized, and it is expected that it will ask for some 120 degree. So here we have just 60 degree in this third member ring, so it cannot breathe properly, right? So that is why this carbocation, it is not stable. So instead, what can happen? So instead, the other mechanism that will happen, suppose the H plus you are placing it here, and this lone pair, it is pushing its electron, and this CC bond will be broken, and as if there is some, suppose I am breaking it, so suppose there is some negative charge generation at this position, so it will, that negative charge will take this H plus. So you can think of it like this. So as if it is generated like this, and then it is taking this H plus. So ultimately what will happen, you will get a CH bond here. So that is happening. Now I have marked the carbons 1, 2, 3, 4 because one of the CC bond that will be shifting. So this 1,3 bond that will be shifting here, and you will be getting 4,3 bond. So this is the newly generated bond, and the 1,3 bond is broken. So final molecule, it is converted to 4-member ring. So that is why if you simply always follow straightforward the normal mechanism, the traditional mechanism, you will end up doing something wrong. So it always depends on the structure of the molecule reaction conditions. We have to be careful about that, right? Next molecule, ring contraction. Again, it is unexpected product. So what is happening here? Let's see. Now the substrate is symmetric molecule, no problem. And here one of the OH you will protonate, and you will get this type of carbocation, right? Now two things can happen. So what can happen? See, if you are simply migrating the pH, so this lone pair, it will be pushing this electron, and pH will be migrating. So when pH is migrating, the positive charge will be generated at this position. So first you can draw this structure, and after resonance, see, this is the carbocation. So this carbocation, it is expected, but it is strained carbocation. So positive charge in 3-member ring, it is unstable. Here also it is unstable because it is expecting 120 degree, but you are not giving it that. So obviously it is now uncomfortable. So it is strained carbocation. So there it is true that there is OH, but still it is strained carbocation. But what is the other possibility? If instead of pH migration, we are doing something else, that is, we are migrating this CC bond. Suppose we are migrating this CC bond, so it will go there. So this CC bond, if it is broken, now positive charge will be generated here. Now see, it can breathe properly. Now there is 120 degree angle possible for this sp2 hybridized carbon, and OH is as it is also present before, and but here strained carbocation, that thing is not present, and pH group, that is also stabilizing it. So it is unstrained carbocation stabilized by phenyl as well as the lone pair of oxygen. So this part is also common, it is also present previous case, but if you simply consider the unstrained carbocation part and it is stabilized by phenyl, then also you can understand that why ring contracted product will be the final product. So simply remove this H, that is minus H plus, deprotonation, we are getting the final product. Next example, influence of reaction condition on the type of product. Now look at the first condition. Suppose it is condition A, this is condition B. So condition A, we are getting expected product because carbocation will be generated at carbon number 1 because P2 pH is there. Now one of the CH3 will be migrating, and you will be getting COCH3, and other side 2 pH 1 methyl. But when we are using catalytic H2SO4 in presence of acetic anhydride, something else is happening. So what is happening here? As you can see, in presence of acetic anhydride, one of the OH group will be acetylated. Now the less hindered OH will be selectively acetylated. So this OH is less hindered, so that is why it will be acetylated. And it is now converted to a good living group. So now what will happen? As it is a good living group, pH will be going there and OSC will be removed. So you are getting PHCO, and then on the other side 2-methyl and pH. Now it has some similarity with semi-Pinacol rearrangement, but that will be part of separate video, okay, not here. So we also have to look at the reaction condition, whether there is any other possibilities or not. Fine. So it is not simply H plus and we are doing the reaction. Here we have acetic anhydride also. Next example, example 6. This is very interesting. Product composition of 4 diastereomeric glycols. So here you can see 2 glycol, that is 1,2 vicinal diol basically. So this is cis diol, right? Suppose this is carbon number 1, this is carbon number 2. So the 2 OH, axial, equatorial, those this is cis, and these two are equatorial, so you can understand this is trans. Now in presence of Lewis acid BF3, it is basically this reaction is also possible in presence of Lewis acid like this. So here we are getting 90% of this product. Now if we take 2 more another diastereomer, now look at this. This is 1, this is 2, this is 1, this is 2. Now this is also trans diol because both the OH, they are axial axial. This is equatorial equatorial. So either you place them as equatorial equatorial or axial axial, both are trans diol. So the green two, they are trans diol, and the other two, that is the blue two molecules in blue cis diol written, they are 1, 2. So see, in the first molecule OH is axial equatorial and now it is equatorial axial. So all the possibilities we have here. Now in presence of BF3, from the first 2 molecules you are getting similar type of observation, 90% of this ketone, and in the second case you are getting 65% of this ketone. Now these two are not same. Suppose if this is molecule A, this is basically A prime, there is a slight difference. Both are ketone, cyclic ketone, fine. But here pH is axial, here pH is equatorial. That is why I have written it as A and A prime. And this is completely different product. And that you can get by ring CC bond migration. This is ring contracted product. Now this is taken from unsolved question. I have mentioned the book Carey-Sandberg, page number is also there, though page number may vary depending on edition. I don't know. No, I'm not sure about that. But if you want to know about the details, reference is also given. So in the reference, though this is a very old paper, but still this is very good paper also. Okay, so you can get the details of it, why we are getting this type of result. But simply we will take the first 2 molecules, that is I am talking about these two, why we are getting 90% of this cyclic ketone, and we'll try to see what is happening here. So one thing is clear that this the products that we are getting, 3 different type of product, A prime and B, they do not reflect the configuration at the benzylic carbon. That means, just let me change the color. So what I am trying to say, if you simply look at these 2 positions, they are actually changed. The OH and pH, they are changed. That means whatever change is there at the C1 position, that is a benzylic carbon position, the 1 carbon is benzylic, right? So they do not reflect, the products do not reflect the configuration. So whatever is the position of OH, pH at carbon number 1, it doesn't matter. You are getting same molecule. And similarly, for the second 2 molecules also, whatever is the position of pH and OH, you are getting same type of product composition, 65-35, right? So it doesn't matter. So only position that matters is C2. So when it is C2, see, this is equatorial, this is also equatorial, and you are getting same type of product. Come to the second pair, here axial, this is also axial, and you are getting similar type of product. So it is reflecting the position only at C2. So for the first 2 molecule, that is a cis diol and trans diol, so what is happening here? Among these 2 OH, obviously this BF3, here water will not be generated. There will be some complex between this Lewis acid and the OH. So it will make some complex here, and there will be carbocation type of that is carbocation, not fully formed carbocation. It may not be so, but carbocation nature, whether it is full carbocation or partial, it will be stable at carbon number 1 because pH group is there. So you will never make complex with Lewis acid with this OH or this one. It will be always at the OH which is present at 1. So this carbocation type of nature. So for the sake of better understanding, I have written this carbocation. So here you will have this carbocation like this. This is sp2 carbon. There will be this vacant p orbital. And when this CH bond is migrating, then this will be migrating from the below position and pH will be the upper position, as you can see. So when you are migrating this H, you will be getting this molecule, right? And if you are considering the other possibility, though here 90% is this and very less amount, around 7 to 10%, though I have not written it in the previous slide. See, I have not written it here, but here also some amount of B is generated. But as you can see, it is generated in the second example in more amount. So that is why I am showing this mechanism with the first pair of molecules, so that you can understand how it is generated. So it is very less amount, but still we want to see how it is generated. When you are migrating this CC bond, then actually it is generated. So in both cases there will be C double bond O, but as the CC bond is broken, you will be getting a CHO at this position. Okay, so details you can get the reference that that I have mentioned. Fine. So that's all about this video. Now when we talk about Pinacol rearrangement, there will be semi-Pinacol rearrangement and also Pinacol type of rearrangement. It is also possible in epoxide type of molecules, but that will be I will be discussing in the upcoming videos. Fine. So that's all for this video. I hope it will be helpful for you, and thanks for stopping by. So that's all.